Everyone, welcome to this week's workshop. Before we get into anything, I want to first talk about a question that we were talking over last week, and there was a slight typo actually here. So in this question, you're trying to determine the suspect of a crime. On the scene, you take a sample from a pool of blood to analyze. Suspect 1 is known to have type AB blood, suspect 2 is type A, and the victim is type O. After doing immunofluorescence sensitive for antigen B, the results showed fluorescence. Who is the culprit and why? So last week, when we were discussing this question, we all talked about how the first step here is to realize that you are doing immunofluorescence sensitive for antigen B, which means wherever there is antigen B in the blood, you will see fluorescence. The mistake that was made last week is in the answer it said suspect 2 because his blood type is the only one that contains antigen B. That was actually a typo. It's supposed to be suspect 1 as you guys had originally answered. And the reason, just to review again, is suspect 1 has type AB blood, and type AB blood will produce an antigen for type A and for type B, while suspect 2 is type A only, which means that they do not produce antigen B, which then means there will be no fluorescence if the blood was just type A, and that's why suspect 1 is the correct answer here. And now to get back to this week's workshop. So this week's workshop will mainly be practice questions, and then at the very end, I will briefly go over some of the concepts in a little bit more detail. So let's begin. The first question is, why is SRP not needed for post-translational translocation? The reason is the function of SRP is to bring the ribosome to the translocon. But if the ribosome is not but the ribosome here is not needed, since in post-translational translocation, that means that the protein is already fully translated. And if the protein is already fully translated, there won't be a ribosome attached to that protein, therefore you don't need an SRP. So for the second question, this is more about integral proteins. So let's say you have an integral protein where the N terminus is in the ER lumen and the C terminus is in the cytosol. Where will these terminuses be after the protein becomes part of the plasma membrane? So the N terminus will be in the exoplasm and the C terminus is going to stay in the cytosol. The key takeaway here is whenever you have a question about directionality of a protein, and by directionality I mean where the N terminus is and where the C terminus is, remember the cardinal rule. Faces are always conserved. And although that doesn't sound like it applies here, it really does. The direction of a protein is always conserved as it goes through its steps in either getting secreted or being put into a certain place in the cell. So the thing here is, if the N terminus is in the ER lumen, it will stay in the ER lumen, and the ER lumen is known as an exoplasmic location, since technically it is outside of the cytosol, and whatever is in the cytosol will remain in the cytosol. What is the difference between hydrolysis of ATP and the exchange of ATP? Now this might be an easier question, but hydrolysis of ATP turns that same ATP into ADP. You have an ATP molecule and then a phosphate group is cleaved off, and now we have ADP. On the other hand, exchange is when an ADP molecule is removed from the situation and a new ATP comes in and takes its spot. And the reason this concept is important here is it's used in post-translational translocation, and we'll come back to how exactly it is used there. But remember, hydrolysis and exchange are two very different things. Let's say you treat a microsome with detergent but do not add a protease. What will a Western blot show us? So remember, Western blots are all about the size of a protein. So that's the biggest hint here. Since there's no protease added to the microsome, there will be no digestion of proteins inside the microsome. Therefore, the Western blot will show one thick band at whatever the size of the protein. Now, the big takeaway to this question, this is really important here. There was detergent added to the microsomes. Why is detergent important? Well, if the same question was asked, but we change this to no detergent added, what happens? Well, what happens here is without a detergent, the membrane of a microsome won't be broken down and the protease will not be able to attack the protein. Let's change this to but add a protease. So without a detergent, the membrane of the microsome won't be broken down and the protease won't be able to attack. And what will the Western blot show us then? Well, it'll show us the exact same thing because we won't have any protein being broken down. Alright, so what is the difference between the hydrophobic regions of a type 1 membrane and of a type 2 membrane? So, in a type 1 membrane, we have what is called a stop transfer anchor sequence as the hydrophobic portion. This portion, what stop transfer anchor sequence means is once this is translated, translocation stops, right? So this, whatever is being translated here will stop, whatever is being translocated, which means moved through this translocon, will stop. That translocation process will halt. And then this hydrophobic region will be transferred into the membrane. So stop transfer anchor sequence involves both the transfer of this sequence into the membrane and halting of translocation of the protein. You see how after this translated protein, this translated portion, the remainder of the protein that's being translated stays out. It won't be translocated anymore. And that's because this is transferred out of the translocon and then the translocon closes. On the other hand, in a type 2 membrane, we have what is called a signal anchor sequence. A signal anchor sequence will anchor itself to this translocon, and then the rest of the protein is still free to be translated, and it'll be able to translocate through this translocon. So big difference here is both do transfer themselves into the membrane, but only in type 2 will we see continued translocation of the protein. In type 1, translocation is stopped. So what role does the charge of amino acids play in the orientation of integral membrane proteins? Main takeaway here is whatever side of the protein you see positive charge, that side will remain in the cytosol. For example, for a type 2 protein, like we were talking earlier, type 2 proteins have a positively charged stretch on the N-terminus side, so we will see this N-terminus in the cytosol. Oppositely, there's type 3, and in a type 3, the positive charge is found on the C-terminus side, which means the C-terminus will now instead be in the cytosol. And an easy way to think about it is, remember that charge makes something polar, right? And so it will prefer to stay in a polar region of the environment. These positively charged amino acid stretches will not like to be pushed through or be in a hydrophobic region. Do translation and translocation occur simultaneously or after one another? So in most cells, they do occur simultaneously. If it doesn't occur simultaneously, we won't see proteins inside the lumen of organelles, right? But in some yeast cells and some higher level, or in all yeast cells and some higher level eukaryotic cells, translocation occurs after translation has already occurred. This is known as post-translational translocation. So, what are the steps to co-translational translocation? Now, this is a picture to quickly and easily summarize this, but let's break this down really quickly. So, the first step is when the SRP binds to the signal sequence on a nascent protein, right? Then this SRP will bring this ribosome over to the SRP receptor. SRP will bind to its receptor, and then we will see hydrolysis of GTP. Now, we'll see hydrolysis in both the SRP and the SRP receptor. What is the importance of this hydrolysis? Whenever this hydrolysis occurs, we will see the ribosome being transferred from SRP to the translocon, and then the translocon will open. Another important thing that I didn't mention is whenever SRP binds to this signal sequence, this protein stops translating. It is not until this step here, when SRP finally unbinds, that's when we see the resumption of translation. Now, as this protein is being translated, the signal peptidase will come and cleave out the signal sequence. And this signal sequence will just float out here, get degraded, and be reused, right? And then translation continues along with the translocation of this protein. So what drives this motion of, what pulls this protein into this lumen? It's actually just the translation of the protein. As more and more amino acids are read, this gets pushed down and then it'll continue to move into the lumen. After translation is completed, this ribosome will no longer bind to the translocon and it'll dissociate. And that is co-translational translocation. So, what is the main takeaway from the translocon experiment? The translocon experiment is that one experiment that used crosslinkers. So the main takeaway here is the protein that is being translated interacts with the alpha subunit of Sec61. Essentially, there needs to be something that this protein interacts with while it's inside this pore, and that something found through this cross-linkage experiment was the alpha subunit. Now, it's important to memorize or to understand the reasoning behind how this experiment is set up, but for the sake of this question, the main takeaway of that experiment is just the alpha subunit is what proteins will bind to. What is the energy requirement difference for translocation in co-translational translocation and post-translational translocation? So in co-translational translocation, as mentioned before, translocation is just pushed forward by translation of the protein, while in post-translational translocation, we actually have ATP hydrolysis of BIP proteins to rapture the protein into the lumen. Now, we'll get back to this about post-translational, and it'll make more sense in the next question. So, what is the process of co-translational translocation? Now, this picture is fairly simple, and let's break it down. So, the first step is a little different for post-translational than it is for co-translational. Since there's no ribosome, since we're talking about a protein that's already translated all the way, the first step will just be a protein with a signal sequence will come to the translocon, right? And once it's through, this signal sequence, again, will be cleaved and removed just like in co-translational translocation. For the second step, now we'll get a BIP protein that is bound by ATP to come to Sec63 and interact. What happens here is once it interacts with Sec63, the ATP is hydrolyzed and it becomes ADP. And once BIP is bound by ADP instead of having an ATP bound to it, it closes on this protein that's being translated. So basically, ATP BIP will be open when ATP is bound, and then it'll come to Sec63 and close as that ATP is hydrolyzed, and it'll close on this protein. Now this process just repeats over and over again as more BIP proteins that are bound to ATP interact with Sec63 and then get hydrolyzed and then bind to it. And this is where we talk about this ratcheting process, as you get more BIPs added and more pulling of that protein down. See how here, this is that first BIP, and by like the third round, it's down here. So we can see how it moves through the translocon. Now once this protein is pulled into the ER lumen, we have an adenine exchange factor. And what this thing does is it takes that ADP that's bound and exchanges every ADP for an ATP. Now this is exactly what we were talking about earlier with the difference between hydrolysis and exchange. Here, instead of a phosphate group just being added to this ADP, we're having this entire ADP removed and then a new ATP being bound. And what this step does is it basically recycles BIP, so then it's ready to come back and bind to the next protein that's coming in. And once you remove all the BIPs, our translocation is complete. So now that we're done with all the questions, I just wanted to go quickly here and talk about co-translational one more time. So we already went through the steps earlier, but I just want to mention that there's some key points that you guys need to know. So the first thing here is the protein is not fully translated. We have, in this situation, what is called a nascent protein that is brought to the translocon, right? And another important takeaway here is co-translational needs GTP for the opening of the translocon, right? We have the hydrolysis of SRP GDP and the SRP receptors GDP. And then, as mentioned before, translocation doesn't require ATP or anything because it's powered by translation. Another important thing here is that translocon experiment that we were talking about earlier. And the main thing here to know is, so you have a translocon, and that translocon is Sec61, right? And the purpose of that experiment was to determine which of the three subunits made the pore of the translocon and interacted with the protein that's being translated. So how did they do this? Essentially, every lysine residue was of a modified protein or modified mRNA, right, was attached with a light activated cross-linking agent. And then once this protein moved through the pore and was exposed to light, that cross-linker would attach itself inside the pore and covalently link that protein to the pore, right? And whenever that complex was isolated and removed and then identified, they determined that the pore was actually the alpha subunit of Sec61. So, as mentioned before, really to simplify this, the main thing to remember here is as a protein is being translocated through a translocon, it interacts with the alpha subunit of Sec61. And then last but not least, key points about post-translational translocation. So we got, in this case, the protein is fully translated before it enters the ER, but it's still unfolded. This protein does not need GTP to come through that translocon because there's no SRP needed. That translocon will actually be opened whenever that protein with its signal sequence arrives. Now, translocation requires ATP in the form of BIPs, and this is found mainly in yeast, but it can be found in some eukaryotic cells. One last thing, actually. This is just a quick slide to recap over all the membrane proteins. So the big things here that you want to know are, for type 1, we have the N terminus in the exo side, right? And exo means ER lumen or the outside. And the reason ER lumen is considered the outside of the cell is because it's not in contact with the cytosol. It's, if you think about it, it's outside of the cytosol, if that makes it easier to imagine. So the best way to memorize these is type 1 is N is exo, C is cyto for type 1, right? Type 2 is the exact opposite of type 1. We got N cyto, C exo. Now type 3 has the same orientation as type 1, which makes it easy to remember because they're both odd. So odd numbered proteins here are N exo and C cyto, right? And then we got type 4 proteins and tail anchored proteins, and then GPI proteins. Now we haven't really gone over this, so I'm just going to talk about tail anchored and type 4. So tail anchored are just like type 2, where C is exo and N is cyto. And the difference here is this C terminal end does not actually extend into the exoplasmic space. Rather, it stays in the membrane, and it's the anchor, as in the name, for this protein. Now, for type 4, the main takeaways here is, if there's an odd number of these transmembrane hydrophobic regions, then you'll have the N on one side and the C on the other side. But if there's even, they'll be on the same side. And that's all for this week's set of workshops. Next week, we can go over anything that you guys have questions over this week. And yeah, have a nice day.